Welcome to the second lecture of Unit 7. This one is titled, If You're Not Growing, You're Dying. We have a quote here from Dr. Albert Einstein. Once you stop learning, you start dying. Our learning goals for this presentation are to understand stages of the cell cycle, define and describe the purpose of the organization of mitosis, which we will define, cite scientific evidence for stages of cell division. Now, we already knew from the previous lesson cycle, Make It Your Own, we know that all cells have DNA from DNA replication. We already know that eukaryotic cells have many organelles and prokaryotic cells have fewer parts, and we already know that things change over time. In our lab, we saw from the onion roots that cells exist in many different states. We know that cells are constantly going through cell division, and that means that if you look at a collection of cells, they're in all kinds of different states, and that some steps take longer than others. So this French phrase, this French idiom, it's pronounced in French raison d'être. You don't pronounce the N in raison. It's not raisin. So the raison d'être, the purpose, the why, is to reproduce, to perpetuate the genome, to pass on its genetic information, the reproduction of the self. That is the raison d'être of the cell. Now, let's start simple. Let's start with bacteria. Bacteria cells are prokaryotes. Prokaryotes are simpler than eukaryotes. They're smaller, they don't have as many parts, they have fewer things going on. That's not necessarily bad, it makes them incredibly efficient. They're very straightforward. They can do things faster and simpler, and sometimes it has a lot of advantages. Now, the way that bacteria cells reproduce is by something that's called binary fission. Binary means two parts, and fission means to split. So what binary fission is how bacteria can split in half into two parts, one becoming two. So a single bacteria will grow. It'll get bigger. It'll get more of itself. So it'll go, more of the genetic material, more of cell membrane, more of all of its pieces. And it'll get bigger and bigger and bigger. It'll go through its DNA replication. When there is enough, when it has plenty of extra, say roughly double its original size, then it will split in two. That way both of the two daughter cells will have enough stuff to survive. So the last one was a drawing, but this is actually a photograph that's taken through a microscope. So this is a bacteria with a crazy name. Let's see, that's Cytokinesis aeruginosus. The pronunciation isn't that critical. I think it's probably Cytokinesis aeruginosus. Anyway, so we see it at three different spots. So on the left, there it is, large. It's almost double the size. Then it begins to divide. You can see there in the middle stage that another cell wall is starting to build to separate them. Then the third stage, there it is separating into two. Now, in binary fission, we end up with two cells that are genetically identical. At least that's the goal. The DNA replication happens first, so you have two sets, and then the cell separates, and it tries to make sure the DNA gets evenly divided. Mistakes can be made, but the goal is that you have the exact same DNA twice. So that's why this says near identical to the mother. So it's a pretty straightforward process. You keep growing, you get more stuff, you get more cytoplasm, you get more cell membrane, you replicate your DNA, the cell elongates, the DNA molecules separate, you start to get division from left and right, a cross membrane starts to form until it completely separates them, and they pinch right apart. So, developing question A1. What would happen if this paramecium, that's what this microorganism is, divided into three parts instead of 2, but without doing anything else different. Everything else is the same for the process. It's just we end up with 3 instead of 2. Well, the resulting cells wouldn't have enough of all the molecules needed. Especially, it wouldn't have enough DNA, because we would have only made 2 sets of DNA and you divide it up 3 ways. There's no way that all 3 cells are going to get all the necessary stuff to survive. Not possible. Okay, proficient question. How many times would binary fission have to occur to go from 1 cell to over 10,000 cells? How many times? Now, the answer here is not 10,000 times, because after the first cell splits, then both the daughter cells will then go through binary fission as well. Okay, so let's try it on a calculator. So we start with 1 cell. After our first binary division, every cell splits in 2. The 1 cell is 2. So there's our first division. The second division, both cells split in 2, and now we have 4. Third, fourth, fifth, sixth, seventh, eighth, ninth, 10th, 11th, 12th, 13th, we're almost there. The 14th division, we cross the 10,000 cell mark. 14 divisions is enough. 14 binary fissions to get 1 cell to be over 10,000. In fact, get 16,384. In fact, there's a fast way to do that, to use logarithms. Log base 2, since we're going to have made 2 go of 10,000, is 13.3 divisions. All right, here's one for you to pause and think about a second before I talk about it. So when eukaryotic cells reproduce, the way that the mitochondria are made and the way that chloroplasts are made is that those organelles go through binary fission. Now, eukaryotic cells don't do binary fission, but mitochondria do and chloroplasts do. In fact, mitochondria even has some DNA inside of it that codes for the proteins needed to make mitochondria. What does this imply about the evolutionary background of how eukaryotes got mitochondria? We can see there in the image there, that's a mitochondria. You can see that line formed right down the middle. It is currently going through binary fission. It is separating. This actually comes from a butterfly cell. What this implies to us is that mitochondria were at one point a separate organism, a separate prokaryote, and it formed some kind of symbiotic relationship with a eukaryotic cell until the 2 species became one, merged. Long process. Now, whenever we were looking at eukaryotes, whenever they looked at it using old technology, the first thing that they could see was that some cells were dividing. They called it mitosis. And there were other parts where it appeared to be waiting, and they called that interphase, meaning the interval between phases. When better lenses were developed and we had access to them, we looked closer, we actually noticed that there was something else happening right in the middle of interphase, that there was a step where it did DNA replication. So we called that the S phase. And there was a gap before and after it, so we had the first gap, then synthesis, then gap 2, which, as scientists, we try to be very efficient, meaning lazy, so we make lots of abbreviations. So that means interphase got broken into two gaps and 1 synthesis, which altogether the three made interphase. We abbreviated them G1, S, and G2. Then, better lenses still, we noticed that the cell wasn't just waiting during gap 1 and gap 2, it was actually going through growth. So it was carrying out its regular cell functions, it was enlarging, copying organelles, things were happening. So still, those three spots all together combined to be interphase. And then the dividing steps we called mitosis. So developing question, which step takes more time, mitosis or interphase? Well, the answer is interphase. That's why we see more cells in interphase than anything else. It takes a lot of time to copy everything. So what data from the lab lets us know which process took up more time? Hint, there were not patient microbiologists with stopwatches. So you take a cross-section of a whole bunch of cells and you look at them during all their different stages and where you happen to bump into them. Well, we happened to bump into more of the cells and interrupted them during their interphase component, which must mean that interphase takes longer. So, in the mid to late 1800s, a German anatomist by Fleming made this word mitosis. It came from the Greek word mitos, meaning thread, and osis, meaning process. He described what he could see, and he thought the DNA looked thread-like to him whenever he's looking at a microscope. You can kind of see that in his drawings, which is where the mitos comes from. And since he kind of did the honors, we kind of kept the name. Actually, a lot of the first cells and a lot of the cells that get studied during mitosis usually end up being embryos. They're the very first cells after sperm joins with egg. Those cells tend to go through division a lot and predictably. And at first, those first cells are quite large. So it makes them really convenient to look at. You get some good pictures. So, mitosis can be broken up into phases as well. So the first phase is prophase. So the DNA takes up a lot of space. We need to organize it. So you put it together in a neat way. By the end of prophase, things will be way more organized. If you took one cell's DNA and stretched it out, although it would be quite thin, it would stretch out 10 feet long. That's for every cell. That's too much. So think about what we do when we have other things like that take up so much space, like long ropes or long extension cables. We need to move them around. What do? We'll wrap them up. It's exactly what we do with DNA as well. So instead of taking like an extension cord and like wrapping it around around our arm and then tying it up, or the way a water hose gets wrapped around those wheel things, in DNA we have this protein called histones. Histones serve just for DNA. DNA gets wrapped around the histones, and in fact, after that, we even coil it up even more, even tighter, into a chromosome. So chromosomes are just lots of DNA that's been bundled up in histones and wrapped all the way up really. And actually, on the very end of the chromosomes, we have this junk DNA that doesn't actually code for proteins or anything. We call it telomeres. Telo means end. Mer means part. So telomeres is the end part. And it's just junk DNA. It just kind of keeps it safe, kind of packs it in. It's packing material. Then we take the two halves and they get joined together. These are our two pieces. They get joined together at a part that's called the centromere. The centromere, centro meaning middle. The centromere, middle part. And it kind of gets pinched in this nice little bow tie like thing. It's, it's not pinched, it's made so that the one side's a little bit longer than the other, so we have the P and the Q part is what it ends up. So overall prophase starts with DNA. It gets wound up around histone. That makes it into chromatin. The chromatin all spins all up real tight, and you get a chromatid. The two chromatids put together, we have two because we have to do DNA replication, so we have two copies. We bind them together with the centromere. So the centromere, the chromosome, has two chromatids from DNA replication. Then we move to the next stage. After prophase, metaphase. So we need to let the DNA out. So the nuclear membrane starts to dissolve, it breaks down, the chromosomes are let out. They attach to these spindle microtubules that stretch all the way across the cell from one side to the other. They attach these parts called centrioles, and they get all lined up so that they're all there, all in a line, ready to go. The spindle microtubules attach on the centromeres on each side of it. These are some actual stained slides of cells going through metaphase. The DNA absorbs the stain very well, so you can see it's the dark purpley stuff there in the middle. You can also see the micro tubules, the spindles that are lined up from one side of the cell to the other that have grabbed on to each side of the chromosome. Whenever we start anaphase, what happens is that the centromeres, the spindle microtubules start to pull. They pull on the centromeres, and the centromeres separate. And so half of the chromosome goes one way, half the chromosome goes the other way. The two sister chromatids, that made up the chromosome that were bound together, half goes to one side of the cell, half goes to the other side of the cell. This is how we make sure each side gets the identical chromosomes, because the centromere bound the two copies together. Now in anaphase, we're pulling them right apart. Here's some actual slides of anaphase happening. You can see how the DNA is stretched out, how it's long and thin. It's following the path of the spindle microtubules, heading in opposite directions, different sides of the cell. In telophase, the chromosomes are all the way fully separated, and then a new nucleus is starting to form. Two new nuclei, actually, one on each side. And in fact, as the membrane for the nucleus is forming, we're also starting to get a new membrane on the edges, a furrow. This little, it's starting to pinch, or cytoplasm is going to start dividing. Here are some pictures of different cells taken through a microscope that we can see telophase in. Some plant and some animal, but you can see the DNA is all the way to one side now. Now it's time to make the full split. So this is cytokinesis. Cyto is cell and kinesis is move. So they're moving apart. So animal cells have a cell membrane, so they just pull together and they pinch right off. Plants don't move and pinch like that, so they just draw another cell wall right between them. The Golgi apparatus walls it off, and they're separated. Here's some pictures of cells going through cytokinesis. You can see the blockier plant cells, those are the cell walls. You can see the wall that's been put up between the two cells. On the right, you can see cells that have a cell membrane. You can see them pinching and completely separating. So if we look at the full cell cycle, we have I, which is interphase, which we know composes of G1, S, and G2, growth, synthesis, growth. And then we go through PMAT, which is prophase, which is where my DNA starts to get coiled up through the histones into chromatids. The two sister chromatids get bound together at the centromeres into a chromosome. Metaphase, breaks down the nucleus. They get attached to spindle fibers and lined up down the middle of the cell. Anaphase rips the two chromatids apart so that half starts moving towards one side of the cell and another identical half starts moving towards the other side of the cell. In telophase, new nuclei start to form, and the cell begins the process of separation, which actually happens and finishes in cytokinesis, ending with two different cells. Okay, so let's try a developing question. And so I'm going to take a second, pause it, think, label. All right, threw you for a little bit of a loop there because I put an extra one in there. So some books and some people like to put a phase right in between prophase and metaphase, which they didn't very creatively name prometaphase. And so basically they just said in prophase we're doing the chromosomes thing, and then in prometaphase we're doing the nucleus breaking down and the centromere thing. It just depends on where you want to, what you want to call what. So interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Back to two new cells that will start going through interphase. All right, well that was a drawing. How about an actual microscope image? So 3 different cells. How about you label these? Well, the one on the left, we can see that it's starting to be pulled apart, but it's not yet. So that's anaphase, the separation being made. The one in the middle there, we can see a full nucleus just hanging out. That cell is not currently separating, so that's interphase. It's not going through mitosis yet. And then the one on the right is telophase. We can see that it's completely divided its DNA, but it hasn't yet separated in the 2 distinct cells. It's about to start going through cytokinesis. Okay. Write a mnemonic. A mnemonic means a memory device. To remember the order of all the parts of cell division. Decide how many letters you want to use. Make up a sentence that uses each letter gets a word. You know, really, the less sense that your sentences make and the sillier they are and the dumber they are, for some reason those things stick in our memory. I don't know, humans are funny like that. A mastery question. This one, let's try some writing skills. How does this process enable the process of evolution? Justify your answer using clear. Stake a claim, provide the evidence that's being used, and then give reasoning. Pause and think about this. Okay, well, the claim is that this process allows us to have the spread of genetic information from our common ancestor that we have with all life. The evidence is that cells can accurately copy all their DNA from one cell and then give it to 2 so that the process moves on, so that the information is spread. The reasoning on how that evidence connects with that claim is that since the information is preserved, since that there's a method that does error correction, that allows the cell to take the information and with very few errors, move that same information to 2 new cells, completely replicating itself in this, kind of self-repairing machine, it causes that same stuff to get moved on and on and on over the generations. That spread of information, that children, daughter cells, have the same traits as their parents is a critical part of evolution.